In this playlist, almost all signaling pathways associated with colorectal cancer will be discussed. In this video, I will discuss the stage 1 initiation of the colorectal cancer, which is caused by mutation in APC, which eventually activates the WNT beta-catenin pathway. So here we can see the plasma membrane and two proteins. One is Frizzle proteins, another is LRP, LDL receptor related protein. The Frizzle protein is actually a 7 transmembrane cell surface receptor, and LRP, which is a co-receptor for WNT signaling. When there is no WNT ligand bound to receptor and co-receptor, the pathway stays off. As a result, the disabled protein, which is a cytoplasmic signaling protein, is inactive. As disabled protein is inactive, then the destruction complex remains active. This destruction complex is formed with three proteins. One is axin, another is APC, and the last one is GSK3 beta. Axin is a scaffold protein which actually holds APC, GSK3 beta, and beta-catenin. So we can say axin is the structural backbone of the destruction complex, and APC, which is a tumor suppressor protein, is the core component of beta-catenin destruction complex. And GSK, which actually phosphorylates the beta-catenin, marks the beta-catenin for destruction when the GSK is active. Now, what is beta-catenin? Beta-catenin is the central effector protein of WNT signaling. When it is stabilized, it enters nucleus and activates transcription, acts as a co-activator, not a DNA binding protein. Now, what actually phosphorylation does? Phosphorylation is the tag that says destroy the protein. After that, the beta-catenin gets ubiquitinated, then it's sent to proteasome and degraded. The result is beta-catenin remains low and unstable in cytoplasm. Now, what is ubiquitin or ubiquitinylation? Ubiquitin are small regulatory proteins that tags proteins for destruction. After tagging, the proteins go through proteasome, which is actually protein degrading complex, which degrades ubiquitin tagged proteins into peptides. Thus, beta-catenin is degraded. In the nucleus, the lymphoid enhancer factor 1 or LEF1 and T cell factor TCF are bound to DNA, but it is paired with Grucho, which is a co-repressor that keeps WNT responsive genes off. As beta-catenin is unstable, it cannot reach to the nucleus and bound to LEF1 and TCF. As a result, the Grucho is still bound to the transcription factors and this signaling remains off. Transcription remains off. When WNT is bound with the receptor and co-receptor, this pathway switches on and activates disabled protein, which actually deactivates the GSK3 beta. As a result, the destruction complex become inactive and the GSK3 beta cannot phosphorylate beta-catenin. As a result, beta-catenin become stable and remains unphosphorylated and migrates to the nucleus and displaces the glucose. Actually, this beta-catenin migrates the nucleus and binds with LEF1 and TCF. As a result, glucose is removed and the transcription of WNT responsive genes are on. Why? This is the initiation event of colorectal cancer. When APC is mutated and becomes defective, the destruction complex cannot efficiently phosphorylate beta-catenin. So beta-catenin accumulates continuously and enters the nucleus and turns the WNT genes all the time. Outcome in the colon crypts. The crypt stem cells escape the growth control. As a result, excess proliferation occurs, which eventually forms polyp or adenoma formation. And this is how the first visible lesion occurs. This is all about WNT beta-catenin pathway. Thank you.